Question. What is the difference between viable and non-viable particle count? Answer. The basic difference between viable and non-viable particle count is the viable particle count, which are at least one living microorganism affected the stability of the product. Non-viable particle count are particles without microorganisms, but they can affect the transportation agent of viable particles. What is the purpose of 21 CFR Part 11? Answer. The purpose of Part 11 is to ensure the electronic record and electronic signature can be trusted or established as much as paper record and link signature. Question. Which guidelines are referred for NVPC? Answer. ISO 146441 Class 1, UGMP Nature 1, USP Chapter number 797, and PDA Journal Volume 57, 2nd March Chapter. Question. Why only 0.5 and 5 micron particle are measured for NBPC? Answer. During non-viable particles count, we measure only 0.5 and 5 micron particle because most of commonly found bacteria are in the size range 0.5 to 5 micron. So the size of range is the main source of contamination. What is difference between alert limit and action limit for NBPC? Answer. Action limit are the generally set through the regulatory guideline such as Nexer 1 that the USP or ISO standard, where the alert limit are specified to the facility should be set based on result of PQ test, and these 10 is historical data. By journal consideration, we don't need compulsory take action when alert level is achieved. It is alert notification to check the compliance state. Question. At which rate air is measured in commercial non-viable particle count? Answer. The particle counter should have a sample flow rate of at least 28.3 liter per minute. One CFU should be filtered as isotonic probe for sampling in the unit flow zone. Question. What should be the isotonic probe location? Answer. The location of the sampling probe depends upon the criticality of the client zone. Sample probe used in the sterile and aseptic environment should be located normally not more than 1 feet away from the working site. The isotonic probe should be within the airflow during filling and closing operation. Question. What is the basic requirement for tube using for particle monitoring device connection? Answer. In the case of portable particle counter, the tube length should be not more than 10 meter, that is 10 feet. Tubing used for particle monitoring shall be compulsory non-particle shading. Sample tube length should be minimized. Although the tubing cannot be totally eliminated. Question. What is the ideal sample length? Answer. It is good practice to take the sample from working height or about 1 meter of the floor. It should be pointed in direction such that the probability of direction of particle is maximized. A poor practice placing the particle counter on the elevator stand or tempo stand. Additionally, it may not be appropriate to locate sample probe directly in under HEPA filter, and it is not recommended to clean room or clean zone. Question. Why particle counter are need to on at least 36 minutes to process monitoring? Answer. Commonly used industrial particle counters working at flow rate of 1 CFM and guideline requirement given on clean room are in cubic meter. To complete 1 cubic meter, we need approx 36 cubic feet air. Hence, we need on particle counter at least 36 minutes prior to process the market. Question: what should be action plan in case of repeat NVPC exclusion? Answer: in case of repeated exclusion, observe that after performing the check and recommendation process in the area shall be stopped. Further investigation shall be performed to identify the root cause. This may include extensive area cleaning investigation, air velocity verification of the HEPA filter integrity testing, airflow studies. Question: why zero count or probe count test done? Answer: before you being with daily sampling procedure, it is recommended to practice false count test. The purpose of this test is to verify there is no internal contamination of the particle counter or electronic problem that would be produce false high count. Question: how we can control non-viable particle counter in clean room? Answer: regulatory focus has been increased using of the portable human operated particle counter. Instead of we can use permanently installed online computer controlled particle counter. Additionally, we can reduce need to operate presence of grade A to place probe to take sample. We can reduce the operator in grade B. We can remove equipment from the aseptic grade, that is large portable counter.